Swayam Prabha. Digital India. Educated India.
Welcome. So we start with our Hodgkin-Huxley equations today. So you learned about the integrate and fire model. Actually, to be more correct, leaky integrate and fire model. And we will change this into the basically the spiking model with using the Hodgkin-Huxley equations. So, as we have discussed, we need now what the incorporation of sodium ion channels, which are voltage dependent, and potassium ion channels, which are also voltage dependent. So, if we think about go back to our circuit again, if we have this I external as before, we have the capacitor, the membrane capacitance on this side, this is the inside, this is the outside, this difference V in minus V out is our V, that is the membrane potential, and we had this G leak and E leak that makes up the leaky integrate and fire neuron. Now, we had said that the path of current is across the membrane, and so that is why we have the G leak and E leak in parallel to the capacitor. And now we also know that the ion channels, the voltage gated sodium channels and the voltage dependent potassium channels, they are also transmembrane proteins. And as you remember from our lecture on ion channels, there is a pore that allows the flow of ion through the membrane. And if we were to consider the ion channels, the sodium ion channels all together, then that is simply going to be an additional branch in this circuitry, a parallel branch. So, what we will have here, if this branch is going to be the sodium channel branch, and then if we incorporate the potassium, that is going to be the potassium channel branch. So, this is because of the sodium. I am drawing it as a box. We will describe what is going to be inside in a little bit. This is for the potassium ion. If we have to have more ion channels, more other kinds of ion channels, which can be again potassium channels, but with different properties, not the kind of voltage gated potassium channels that we will be using here, then those will be additional parallel branches that can be added on here. We can have calcium channels, we can have chloride channels, and so on. So, as we have been mentioning, that to explain the behavior of action potentials, the sodium and potassium channels are sufficient, and that is what Hodgkin-Huxley did in their experiment with the squid giant axon, where they did patch clamp experiments on the squid giant axon, and with a variety of measurements came up with the exact properties that govern this circuitry, with the properties of what the properties of these two boxes should be. So, let us start considering just by the sodium channel branch, and so what what we have in this branch, what we should have in this branch is that because of the flow of the sodium, there is essentially a conductance present for the sodium ion, and this conductance is variable. So, I am drawing this block here. So, we have what we can call that conductance is GNA as a function of the voltage which is across the membrane. This is if you recollect our discussions on the voltage gated channels, this is this essentially must capture the behavior of the ion channel as the voltage changes throughout when there is some current injection. Now, as you know that sodium ions have some equilibrium potential based on the concentration of sodium inside and outside the neuron, and that equilibrium potential or reversal potential is, let us say, ENA. So, sodium reversal potential. So, what we mean by reversal potential, just to recollect what we had studied early on in this course. So, it is if the membrane potential is at ENA, that is V is at ENA, then there will be no net sodium ions flowing through the membrane. Or also, if the voltage is different from ENA and there are paths for sodium available to flow through the membrane, then the sodium will flow in such a direction so that the voltage is pulled towards ENA. Similarly, we will have the same thing for potassium, its reversal potential, and so it is dependent on the potassium concentration outside and inside. So, given from our previous lectures, this ENA is close to plus 40 millivolts and this EK is close to minus 85 millivolts. These are approximate values, they also can vary, but when you will be actually calculating those, you will be provided with the actual concentrations to be able to calculate them. So, for now we will assume these are the nearby values to understand the system. So, now as we introduced the idea of EL earlier, similarly here we will have now ENA, because if this voltage V across the membrane, or in this case here this V is equal to ENA, then there will be no flow of current through this membrane, because the current through this branch is essentially GNA V into V minus ENA, where GNAV is the sodium channel conductance as a function of voltage. And so, if V equals ENA, this I sodium is 0. So, here now we have the new components INA, that is I sodium, and IK or I potassium, and we have our old I leak and I capacitor. So, similarly, the branch for the potassium is simply going to be our GK times is GK as a function of V followed by this battery EK. So, now if we draw the full circuit and write out the entire set of equation or the entire equation, we have, this is our I external, we have the capacitor C, we have GNA as a function of V followed by ENA, then GK as a function of V followed by EK, and this is G leak, which is passive, which are not the voltage gate, which is not like the voltage gated conductances, and this E leak, which stops the flow of the ions along those branches, that branch, sorry. So, capacitor. So, and this is inside and outside. So, exactly in the same way as we did for the leaky integrated fire, if this is INA and this is IK, we have I external is equal to I capacitor plus INA plus IK plus IL. So, our I capacitor turns out to be I external minus INA minus IK minus IL, and so we have C dV dt is equal to the I external, then minus GNA V minus ENA minus GK V minus EK minus G leak, GK function of voltage, and V minus E leak. So, in now, like we were earlier saying, we would simulate this differential equation with a starting point, and then see how the voltage changes with time for a given I external over time. We will have, if we enforce a V threshold, we can find out where the spiking is going to happen and so on. But in order to do that, in this case now, we have a very difficult problem, and that is, what is the nature of this GNA and GK? That is what Hodgkin and Huxley, with their immense amount of work and insight, showed us how to model the GNA and GK, the voltage gated potassium conductances and voltage gated sodium channels. So, if you recollect, when we talked about the patch clamp recordings, we had done this little experiment that was similar to what Hodgkin and Huxley did. I mean, we had discussed the experiment, that is, we do a voltage clamp, that is, we are at V rest. This V rest is, let us say, minus 60 millivolts, around there, and we go up to minus 20 millivolts, and we clamp it there. This is our V clamp, and we bring it down some 10-15 milliseconds later. Now, if you remember, with variety of other information, by putting in cesium ions, and that blocks the potassium channels, and now we are left with only the sodium channels. If we assume that it is only the sodium and potassium channels that are involved in this, and actually it turns out to be sufficient for the squid giant axon. So, for this we show that the current that we get, that we measure, goes up or in magnitude and then goes down and stays at 0. So, this is 0 current, this is the I axis, and this is the time. So, at this particular point, there is an inward current, that is positive ions going into the neuron, which is why this is negative sign in our convention, and then it goes back up to 0. And again, similarly, on the other side, if we block the sodium channel and plot the potassium current with the same kind of voltage clamp, we get the potassium current to increase and saturate at a particular value until the clamp is held, and then it goes back down to 0, I 0. So, these two observations actually tell us how to go about or start thinking about modeling these GNA and GK. So, let us say somehow we have blocked these sodium channels, and we know because of the integrate and fire model, this G can really, we can have measurements to G to get G and EL, GL and EL, and so that can be that is not a problem to estimate. Then what we have here is, if we remove this from this equation, we have C dV dt equals I external minus G k as a function of voltage V minus E k. So, if we can measure the currents during this period where the sodium channels are not working, or if it is closed, if we can manage to close them, this is in here, it was with TTX, as you may remember from our previous lectures. So, what are we doing essentially in this experiment? We are clamping the voltage at a particular value, which means that our C dV dt is set to 0, because the voltage is fixed, it cannot be changed. Experimenter is controlling the voltage exactly at the particular V that we want, that the experimenter wants. So, if this is 0, by measuring the current, we can easily get basically I external is what is the profile of the GK into V, GK as a function of V minus EK. And so, by using different clamp values, let us say this is V rest and back to V rest here, and we clamp it to some V1 here, we clamp it to some V2 here, some V3 here, some V4 here, some V5 here, and so on. So, essentially what we are describing, each of these are different experiments, just like what we did here, only the voltage clamp value, the clamp value is different in the different experiments. So, in each case, for the voltage clamp, in each case for the clamp value to be V1, V2 and so on, we are measuring our I external is equal to the GK as a function of V minus EK. So, we know what EK is, we know what V is, it is in our control, and we are measuring I external because of the patch clamp. So, from that profile, what we are essentially we can determine is how GK is changing over time when we fix the voltage at a particular value, which may be V1, which may be V2, which may be V3 and so on. So, what we will see if this experiment is done, if the potassium current by itself, if this is, let us say, we will have each experiment for a clamp at V1, V2 and so on is described by the successive lines here. So, at the lowest V, what we see, so this is time, what we see is, and we are having a clamp for this period, and this value is our V clamp, which is our V1 etc. That is, this case is for V1, this case is for V2 and this case is for Vn. And the current is the IK or I potassium, which we will see is increases to a particular value and stays there till the clamp till the clamp is removed, and it drops down and goes back down to 0 current. So, this each of these is 0 current, and this axis is current. If we go, so this is for V1. Now, if we increase the voltage to some other value, we will see that it is increases more. Then in the next case, we will see that it rises and goes back to a high value even faster, and on a larger value too, and then even larger value and drop back. So, as you can see, first of all, with increasing voltage, so this is V1, V2, V3, V4, the largest value of the potassium current and hence the conductance at that voltage keeps on increasing, and that is as a function of voltage, the largest value of the potassium conductance keeps on increasing and then saturates at a particular value. The other observation here is that mostly in this description, we have a speed or a time constant that governs the rise of the potassium to that particular voltage. So, this time constant turns out to be decreasing as the voltage increases in this particular depiction of the potassium currents. And so, from these we can actually, if we were to fit let us say an exponential, we can derive a time constant or know what the time constant is for each of those cases or each of those voltages. So, as a function of voltage, if the maximum G N A that it reaches is of this kind, then we have one particular depiction of the behavior of G N A as a function of V. Remember, we have said that this G N A as a function of V needs to be completely described in order to solve that differential equation that we had, which is our C dV dt equals I external, and then we have minus I N A minus I K minus I leak, where we are now looking at this I K, which is described by this G N A into V minus E K. Sorry, not G N A, G K into V minus E K. So, and here also we are talking of G K. I am sorry for that. Now, so we need the entire dynamics of how the G K is changing with V. What this experiment has provided us so far, at least, is the value of G K. I mean, we can indirectly infer the value of G K as a function of V, but what the G K value is going to be if the potential is held at V for a fairly long period of time. That is, if I am holding the voltage at a particular value, let us say we were at minus 60, which is the resting membrane potential, and then we hold it at minus 40. And if we hold it for a period of time, then the potassium conductance finally goes up and to a particular value and stays there for a long time. It saturates there. So, in a sense, what we at least have is what the value should be of G K when the potential is when at steady state, essentially. So, if we can measure how this is changing over time to that value and incorporate that dynamics into the G K, then we would be able to solve the differential equation, because simply the final value of Gk at that clamped voltage is not sufficient for us to solve this equation to get the voltage across as a function of time. So, in order to do that, what we need is to obtain the time constants here. Similarly, for the sodium currents again, we see that if you talk that if we have the sodium currents here, it is small here actually. Similarly, in parallel here, if we are plotting the INA and correspondingly the at the different V's that we have at different times, then what we will see is that there is a increase in the magnitude of the sodium current, and it reaches its final values faster and faster. So, there is a increase in the magnitude as a function of voltage, and that there is a hastening of reaching the peak value there. So, again from here also we can obtain some sort of a time constant. But the big difference between the two, as we had mentioned, is that there is a component that brings the current back to 0. And so, with these two observations, what Hodgkin and Huxley essentially proposed is that the potassium channels, the potassium ion channels, have only activation gates. That is, once the channels are, as the voltage increases, the gates open more and more. The number of gates over the probability of the ion channels, the potassium ion channels being open keeps on increasing. We will get into the why we said probability in the next lecture, but for now let us think of it in this way, that more and more potassium channels are going to be open as we increase the voltage. On average, more opening of potassium channels as voltage increases. But in terms of the sodium gates, sodium channel, they propose that sodium will have activation gates as well as inactivation gates. So, what we mean by inactivation gates is that if the voltage increases, the ion channels, the sodium ion channels starts to close. These particular gates start to close. That is, as a function of voltage, if we think of the probability of opening of inactivation gates, then as a function of voltage, they keep on dropping, they keep on reducing as the voltage increases. Where it goes, those particular details we will be covering in our next discussion on the Hodgkin and Huxley equation. So, here so far what we have shown is that experimentally the sodium conductance and the potassium conductances as a function of voltage clamp, that is different values of voltage clamp, those can be obtained and their profile over the duration of the clamp can be obtained. The difference between the potassium and sodium is that potassium goes and reaches the saturation and stays there until the clamp is removed. The sodium goes and reaches some value and then the current or the conductance goes back down to 0. And what Hodgkin Huxley proposed, that this can be modeled by assuming that potassium channel opening is controlled by gates that are activated by voltage, that is, or activation gates, that is, with increasing voltage more and more of those gates will open. And sodium channels have two types of gates, activation as well as inactivation. One that behaves like the potassium channel gates and another that behaves in the opposite way, that is, as voltage increases their closing chances keep on increasing, or their opening increases with decreasing voltage. So, with these ideas, we will start off in our next continuation lecture of the Hodgkin-Huxley equations and see how there will be a set of differential equations that will come about by incorporating all these ideas and show action potential behavior. Thank you.